The impacts of high-intensity grazing on land is manifold. Soil is degraded and compacted through trampling, decreasing soil productivity and fertility. Without proper growing conditions for vegetation, this bare soil is vulnerable to erosion and eventual desertification. This process can have a global impact. Arid ecosystems are estimated to cover 1/3 of our planet, and soil is known as one of the world's largest carbon sinks. However, with erosion becoming more and more prevalent through livestock grazing, carbon is being released and acts as an accelerating agent to global climate change. Anderson Mesa is about 20 miles southeast of Flagstaff, Arizona. An experiment was conducted starting in 1997 to study the effects of livestock grazing on grassland. The area was sectioned off into different transects, with some sections receiving high-intensity livestock grazing and some receiving none. Various aspects of the ecosystem have been monitored since then to measure the impacts of high-intensity livestock grazing on land. Our team's hypothesis based on the Anderson Mesa experiment states that high-intensity grazing causes less vegetation than non-grazing methods.
The importance of analyzing plant cover and vegetation was to determine if there were changes between the pasture environments at a localized point. To measure these differences, we measured the cover of graminoids, forbs, shrubs, litter, rock, and bare ground. We performed this analysis using the quadrant method. This method involves using a meter square and approximating the amount of individual species, bare ground, and other noticeable features within that square near our 3 dig sites. Every group member agreed upon the percentages of species and plant cover, but this data was not exact and percentages should be expressed with error. We also recorded the tallest plant height using a ruler and recorded the different heights.
We did a couple of tests to identify the soil on our plot. First, we used a soil probe to test the compaction of soil. Then, we wet the soil, identified textures using our hands, and used a flow chart to figure out what kind of soil we were handling. Lastly, we collected soil samples to bring back to the lab to figure out how much moisture was in the soil. We did this by comparing the initial weight of the samples to the weight of the samples after putting them in an oven and completely drying them out. We were also interested in seeing if grazing had any effect on insect populations. First, we did a sweep at each point along our transect line, making sure to do 10 sweeps at each spot. We collected the insects found in the sweeps and brought them back to the lab. In addition, we set up insect traps, one at each point along the transect line, so we could trap insects and come back to collect them in a week.
Having a sense of place in the area that you are studying is important because it allows you to appreciate and feel connected to the land. To do this, we sat quietly for 5 minutes to hear, smell, and feel everything that was around our site. Next, we named the place that we were in and gave it a story based on what we saw and felt. A geographic information system is a device that captures and stores geographical data, which can then be used to identify trends in the data. We use the GIS system to map all transects and the corners of our plot. These data points can be used to relate the data taken from the plots and the physical locations they were taken from. The air temperature and relative humidity of the plot are important because they can impact soil moisture or plant growth. It was 70 to 75 degrees Fahrenheit on the day we were on the site. To find the relative humidity, we used a device called a sling psychrometer, which had a wet bulb and a dry bulb and was flung around in a circle above our head for a minute and a half. The wet bulb read 68 degrees Fahrenheit and the dry bulb was 71. These two temperatures meant that the relative humidity of the area was 42%.
After collecting and comparing data from 36 transects in 4 grazing plots, there is evidence that shows that vegetation on Anderson Mesa is not significantly affected by various grazing methods. Of the calculations completed thus far, the most notable difference between grazed and ungrazed plots is the percent of land covered with forbs. Average plant height, percent plant cover, and bare ground were relatively equal. Additional graphing calculations would be necessary to discern a pattern in the data.
Our original hypothesis stated that high-intensity grazing causes less vegetation than no grazing method. However, through various experimental methods and collection of data, our hypothesis was not supported. It has been shown that grazing methods have little to no effect on plant cover.
In general, no substantial correlation was seen in regards to plant cover and the method of grazing used on the plots. It is possible that the reason the plots did not see a significant change in vegetation cover was due to other factors that our experiment did not take into account. To prepare for future experiments, more research on how the plots function regardless of the grazing method would need to be done. This would allow for the experiment to be more controlled in what variables are responsible for what, since the core understanding of the land will be increased. Another reason that the results were surprising is that in each of the individual research papers we found Alan Savory's claims were largely inaccurate.